Welcome back, everybody. This is going to be a fat one, so lean in, turn up your volume, get ready to do it. I'm going to skip the recap again. We're talking about recombinant and complementary DNA in this video. Okay, so we're looking at plasmids and how we can edit the DNA in plasmids. Okay, again, we're now moving on to this dot point down here. So the use of recombinant plasmids is what we're going to look at, and vectors to transform bacterial cells as demonstrated by the production of human insulin. All right, we'll get to the insulin next video. All right, so what is a recombinant plasmid? All right, so bacteria, as we know, they have a few different things in their anatomy, all right, that we should be well aware of at this stage of year 12. Okay, obviously they're a single-celled organism, so they're a eukaryote. All right, so they don't have nucleus. They don't have a nucleus or membrane-bound organelles, but they do have organelles such as ribosomes. All right, and they have this circular DNA, which I like to call spaghetti DNA. But they also have these circular, like pure, perfect round circles of extra DNA, which they call plasmids. Okay, and that's really what we're looking at today. Okay. So once a plasmid is edited to integrate a target gene, it is referred to as a recombinant plasmid. So if that's been edited and a new gene's inserted into a plasmid, we call it a recombinant plasmid. And that's what we're looking at today. So something to note about plasmids is that they can be referred to as a vector. Now, what's a vector? It really is a means of introducing foreign DNA into an organism. All right. There's lots of different things we can call vectors. Plasmid is a vector. A virus is a vector because it is just injecting foreign DNA. A tick or a mosquito, for example. So a mosquito gives you malaria, but how does it do that? It injects foreign DNA in the form of a plasmodium. Okay. So yeah, all right, can be referred to as a vector. It carries foreign DNA, can put it into something else. So plasmids are excellent cloning vectors because they can self-replicate, all right, are small and can be taken up by bacteria. So this is really the perfect way that we can actually edit genes, okay, using vectors, such as plasmids. All right, here's where the thick of it really starts. So plasmid vectors, once they've been edited, they're going to contain a few different things. Okay, so it might look like a lot, but I want you to look at the images below. Okay, as we go through this. So the first one here is the restriction endonuclease site, and we learned last class, that the restriction endonuclease is the molecular scissors, and it's going to cut at a particular site. Okay, so you can see that here in this example, the BAM-H1 is your restriction endonuclease site. Okay, here. So that can be cut here. All right. There's going to be another site, echo R1, that can be cut here. Okay, those two cuts then form this area here, which something can then go into in the plasmid. All right. And the thing to note is that those cuts are going to be sticky end cuts. All right, so again, remember that's the Z-shaped cuts that we looked at. So they're going to look something along the lines of that with your nucleotides in there, your bases in there. Okay. So yeah, so just keep that in mind, and you'll be able to see that. As you can see, it's that Z shape, the cut. BAM-H1 or echo R1 is that Z shape, that sticky end cut. And that's easier to put back together. That's why it's a sticky cut. Okay. Through the form of the H bonds between the complementary base pairs and DNA ligase connecting the bonds between each of the nucleotides there. Okay. Next, we have the antibiotic resistant gene, and this is essentially it's one way that we can test whether a bacterium has taken up the plasmid. Okay, because they don't always get taken up, but we'll learn more about that in the next class. All right, so for example, in this one, we have an antibiotic resistant gene here. All right, you can see that in the green. All right, so an example of that would be an ampicillin resistance. All right, so as you can see, ampicillin, that's the antibiotic, and then this superscript here with the R, that's resistance. All right, that's just denoting that it's the resistance of ampicillin. We have this marker called an origin of replication. It really doesn't, we don't need to know what that does, all right, but it is going to be there in some examples, all right, so that's shown in the yellow, all right, sequence to show this. Cool. And then lastly, we have the reporter gene. Okay, so where is that going to be? That is where we're cutting, all right, so you can see that here, the black part of the plasmid. All right, that's your reporter gene. As you can see, it's perfectly functional here in this example, but then it's been cut twice with restriction endonucleases. All right, so it's been opened up, and a gene of interest has been inserted. So then it's not going to work anymore. Okay, so that will not work once a target gene has been inserted. All right, but what will work, and by work I mean what the bacterium will express in its proteome is actually the gene of interest. Okay, you can see the gene of interest here in the blue. All right, so reporter gene, easily identifiable phenotype that can be used to identify whether a plasmid has taken up the gene of interest. Okay, so for example, actually we're going to see that on the next slide. Okay, so we'll go into that. But yeah, and obviously in the blue you have the target gene of interest, which has been cut out of some sort of eukaryotic organism or potentially a prokaryotic organism, and it's going to be put into the prokaryotic plasmid here. Okay. So the basic process of editing these plasmids or recombination is essentially you have your plasmid, you cut it open with restriction enzymes or restriction endonucleases, we should refer to those as. You stick your target gene in, hopefully it joins up with DNA ligase and the sticky end cuts. All right, and there you go. If that works successfully, tick, you get a recombinant plasmid. All right, sometimes it's not successful, sometimes it is successful. That's the basic process of that. So what is required? Okay, so again, we're just going to look at the same things we looked at before, but we're going to add in some of the things that we learned about last video. So in order for scientists to create a recombinant plasmid, they require a gene of interest, obviously seen here. All right, this is what was shown in the blue before. All right, just a gene that they've taken from a eukaryote, and as you can see, it notes that there's no introns. Okay, so how would we get a piece of DNA from a eukaryote without introns? You should be able to answer this based on what we've learned last class. I'll give you a few seconds to figure that one out. Okay, that should be enough time. So essentially, we get the RNA of the specific target gene. Okay, and we use that reverse transcriptase. Because remember, reverse transcriptase will then essentially recreate the other strand that's missing from the RNA, but obviously the RNA can have the introns removed during RNA processing. So in other words, you get DNA with no introns, which means it's then compatible with the bacterial or prokaryotic DNA because they do not have introns, because they don't have that step of RNA processing. Okay, so it's all coming together. The puzzle pieces should be coming together. Cool, so we have that gene of interest without introns. We have a plasmid vector, so we just have a plasmid here we can use. All right, we have a restriction endonuclease, which is going to cut at some point of the plasmid, hopefully within that reporter gene that we spoke about last slide. And then we need DNA ligase as well. All right, so if we look at the plasmid itself, we have random DNA. There will be an origin of replication in here somewhere. So say it was just that part. All right, not really that important, but it would be there. All right, we have our resistance, some sort of resistance gene. Okay, so this time it's tetracycline, antibiotic resistance. We have our green fluorescent protein. Now, what is that? You essentially take that out of a fluorescent jellyfish and you stuff it under your Christmas tree. All right. In other words, you take that from another organism and you will put that into your bacteria and they will end up glowing green. Okay. And that is your reporter gene. Obviously, we have our restriction site here, as you can see, and that's going to be the spot where our target gene slots in. Okay, now the yellow's not looking too nice, but that's where the target gene is going to slot in. Okay, very good. So let's look at that now. So what we want to happen, the restriction endonuclease spliced gene of interest may fit into the spliced plasmid. Again, splice just means cut. All right, we have those sticky end cuts you can see with that Z shape. Obviously, we want this to go into here. All right, sometimes it does, sometimes it doesn't. All right, remembering that the bases here are going to be complementary with the bases here. All right, so those H bonds are going to be essentially reconnected thanks to the sticky end cut, and thanks to ligase, the phosphodiester bonds are going to be stuck together again. Okay, so if I was to color code that on this plasmid here, I'll try and do it in a color you can see. So I'd put the H bonds in blue. H bonds will be here, all right, like across that side. If you were to put like stitches, it's where the nitrogenous bases are complementary. All right. Whereas the DNA ligase, I'll put that in. I guess I'll just do black. All right. That's going to be across here. Okay. So that's going to be more horizontal on the strand. All right. And that is, might be difficult to see, but that is something important to note. Okay. And that is the difference between the DNA ligase and its job and the H bonds that are just going to be sticky to one another, right? Hence the sticky end cut. So that's what we want to happen. We want this gene to slot in here and then be incorporated in the bacterial genome and therefore bacterial DNA, and therefore whatever the gene is will be expressed by the bacterium and will be produced into a protein, a working polypeptide. All right, so finally, how can this technology be used? All right, it creates valuable substances or molecules by making the bacteria express the genes we want. All right, as I just mentioned, if the bacteria express those genes, that means the proteins are made. All right, once those proteins are made, we can do whatever we want with them if we are able to extract them, which we can in certain scenarios. Okay, perfect. So the perfect example is creating human synthetic insulin. All right, so human insulin. People that are diabetic, they need this. All right, how can you create this using the bacteria? All right, such as Escherichia coli, E. coli. You essentially just do exactly what we learned today in this video. You get your human cell, so eukaryotic cell. You take the gene well, really it's two genes that you put together, but anyway, we'll learn that next video. You take the gene that creates insulin from human cells, all right, and that is your target gene. You take a plasmid from a bacterium, preferably E. coli because it replicates quickly, and you insert that insulin gene into the plasmid to make a recombinant plasmid. All right. Then you do another process of which we haven't learned today, but we will learn next class, next video, and you put that plasmid into the into the bacteria to make a transgenic bacteria or a recombinant bacterium. Okay. Hopefully that will not die, because some of them die, some of them don't take up the plasmid, but then some of them will. You find out which ones will using the reporter gene, among other things such as the resistance, all right, the antibiotic resistance. Then you grow those in a culture once you've isolated the particular transgenic bacteria. And from there, they're going to produce those proteins. So they produce the synthetic human insulin protein. You extract that, you mix it together, and kaboom, there you go. You have human insulin that you can inject into a person, and it does the exact same job, the exact same role, because it is just a functional polypeptide as insulin that's produced in your body, in your liver, for example. Okay, and that's it. That's today's class. Hopefully you learned something from today. It's a big one. You might need to watch this a few times. Please like, subscribe, and comment. And good luck with everything in year 12. Good luck for your SAC coming up soon. Thanks, guys. Have a nice day.